What is the gene pool and how is there so much diversity in it? How do mutations occur and what can they lead to? Well, the gene pool is all of the alleles that exist within a species, but how can these alleles change? My name is Alyssa, and in this video we will investigate these and other questions to help you gain confidence on test day. We'll start by looking at the different kinds of mutations and then look at what the consequences of these mutations are. We'll finish off by examining two different terms, genetic leakage and finally genetic drift. After watching this video, you will be able to identify the different kinds of mutations and recall how they affect the organisms in which they occur, and then you will be able to answer questions that relate to this topic. First, we will investigate the different kinds of mutations that can occur at the nucleotide level. These mutations can be a result of chemicals or of radiation, both of these terms called mutagens, or from mistakes in DNA replication. Many mutations that occur are point mutations, changes in one single nucleotide in the sequence. Here's an example of a DNA sequence coding for a specific protein, with each codon, or three-letter sequence, matching the code for an amino acid. What happens if one of these nucleotides gets changed? In this case, guanine is present instead of alanine. But the amino acid matching the code is still leucine. That is because amino acids have more than one codon to code for them, and typically the difference between these codons is found in the third nucleotide. This is known as a silent mutation because it did not affect the final protein and therefore isn't really noticed. It most commonly occurs when the mutation is at the third letter of the codon because of the degeneracy in the genetic code. What happens if the mutation is at the second nucleotide of the codon? Here, a cysteine replaced a thymine, and instead of coding for leucine, this three-nucleotide combination codes for serine. This is known as a missense mutation because now the protein is affected by the mutation, and chances are it won't function exactly as it should. What about if the nucleotide that is changed makes the codon a stop codon? Here, guanine has replaced thymine and has resulted in a stop codon. The protein is now severed at this position when it should have grown longer. This is called a nonsense mutation because now the protein is incomplete and there is no way that it will function properly. The last nucleotide mutation that we will look at occurs when a nucleotide is completely removed from the genetic sequence, not replaced. In this case, a thymine nucleotide has been removed, and the whole genetic sequence has shifted over and is one nucleotide shorter. This has altered not only which amino acids are added to the growing protein, but has also coded for a premature stop codon. This is called a frame shift mutation, and in this case, a 1 base pair deletion has occurred. Sometimes a nucleotide can be added to this sequence, and this is called insertion. Both insertion and deletion cause grave changes to the protein, but if 3 base pairs are added or deleted, the severity of the mutation is lessened. This is because a codon contains 3 nucleotides. When 3 nucleotides are added or deleted, the protein can be synthesized to completion unless a nonsense mutation occurs. Note that this protein will have many different amino acids than the normal protein, and this may affect its function. Now we'll take a look at the mutations that occur at the level of the chromosome. In these cases, large segments of DNA are affected, not just individual nucleotides. When a large segment of DNA is lost from the chromosome, it is known as a deletion mutation. Initially, this segment is found on the chromosome, but somewhere along the way it has been removed. This can cause problems when the information lost is important for physiological and biological functions. Small versions of this are the frame shift mutations that we just examined. Duplication occurs when a section of DNA is coded multiple times throughout the genome. This leads to duplicates in the synthesis of the proteins relating to that section, which may or may not cause problems to the individual. Inversion occurs when a segment of DNA has been flipped in the opposite direction. This typically does not cause any abnormalities in the individual. There are also insertion mutations, where a segment of DNA from one chromosome is moved to another and inserted into that chromosome. Here, one segment of the yellow chromosome has been deleted and was added to the gray chromosome, making 2 new chromosomes. Small versions of this are again the frame shift mutations that we discussed earlier. Finally, there are translocation mutations. In these mutations, segments of DNA are exchanged between 2 chromosomes. So in this case, a piece of the green chromosome attaches to the red chromosome, and a piece of the red chromosome attaches to the green chromosome. This again creates 2 new chromosomes. We know that these mutations can occur, and we talked a little bit about what happens when nucleotides are mutated, but what are the overall consequences of mutation? Let's take a look at the example of the peppered moth. In the first image, the light-colored moths are blending into the light color of the tree, and the dark-colored moths resulting from some genetic mutation are sticking out like a sore thumb. These poor moths are easy prey for any birds who are flying by. Therefore, there are only a few dark moths and much more light-colored moths. But in the second picture, we see that growing industry in that area has caused the tree bark to darken from all of the soot. In this case, being a dark-colored moth is beneficial because they can now blend into the bark and the birds will prey on the light-colored moths. Here we can now see that there are more dark moths than light moths. In this case, the mutation that caused the dark color is an advantageous mutation to have, as these moths are surviving and creating offspring under these circumstances. Though we would like to see only advantageous mutations, there are cases in which mutations can be detrimental. Most often, this is due to the inability to form functioning proteins. In the case of sickle cell, having 2 copies of the mutated allele causes individuals to have distorted, sickle-shaped red blood cells due to issues with the hemoglobin molecules. This produces a number of health issues for individuals suffering from this disease. However, this mutation can be beneficial if only one allele has the mutation, because in these cases, individuals are less likely to die from malaria. We will now discuss genetic leakage, which occurs when genes from one species are combined with genes from another species. An example of this is Agrobacterium. This bacteria can transfer its genetic information to plants, which causes the infected plant to grow tumors. This bacteria is now being studied so that it can insert specific genes into plants and cause advantageous changes to the plant instead. In other cases, species that are closely related will mate and produce what is called a hybrid. The hybrid itself is an example of gene leakage, the net flow of the parent's genes into this new species. If this hybrid has an even number of chromosomes and is able to reproduce, it will cause gene leakage again when it mates with an individual from either parent species, and the genes are passed along to the new hybrid. Lastly, we will explore genetic drift, which is defined as the changes in the gene pool that occur just by chance. Remember that the gene pool is all of the alleles that exist within a species. Genetic drift is more pronounced in smaller populations because there is less opportunity for mating outside of genetically related individuals than when there is a large population with genetically different individuals. Take for example this population of red and green organisms. Originally, the frequency of red and green organisms is 50-50. However, if some sort of event or natural barrier is put into place that isolates this portion of the organisms, genetic drift is more likely to occur. If these organisms continue to reproduce with these barriers in place, we can see that more and more red are present, and eventually they consist of the entire population. This occurs simply by chance because of the way in which alleles combine when forming organisms. Genetic drift can be detrimental to the population because it leads to less variation in the gene pool and can cause reduced fitness of the population. This example using the red and green organisms is an extreme case of genetic drift and is known as the founder effect. The founder effect occurs when species are in reproductive isolation from other populations. Genetic diversity therefore decreases, and the prevalence of certain traits and even diseases increases. Therefore, you can see why it would be beneficial for some mutations to occur in this case, or to have increased opportunities for outbreeding, as these would provide options for increased diversity in the gene pool. Now that we are a bit more familiar with the topic, let's do some practice questions to test your skills. This question is asking you to think about the different kinds of mutations and what potential consequences they could have. The answer is D. A single nucleotide insertion early in the mRNA sequence will still yield a normal, fully functional protein is not true. This will result in a complete frame shift. The 3-letter codons will be read incorrectly from the point of the insertion, thereby coding for incorrect amino acids thereafter. This will not yield a normal protein that was originally intended. Answer choice A is true because in translocation mutations, all of the correct genetic information is there, but it is just in the wrong place. It is possible to see no harmful effects from this mutation. B is true because nonsense mutations mean a stop codon was inserted, resulting in termination of translation, a truncated protein. Finally, C is incorrect because by definition, silent mutations are silent in their effect. A different nucleotide may have been inserted, but because there are multiple codons that code for the same amino acid, you will still have the correct amino acid inserted into the protein chain. This question is asking you to think about the causes of the increased prevalence of a certain trait. The answer is A, the founder effect. Remember that this occurs when a small portion of the population becomes reproductively isolated and similar traits are passed throughout the population. This is an extreme type of the genetic drift we discussed earlier. Answers B and D are incorrect because these are random mutations that have harmful effects on the individual, and therefore reproduction to pass along the trait is unlikely. And C is incorrect because outbreeding introduces genetic diversity into the population, and traits would therefore not be similar. Fantastic work if you got those questions correct. In this video, we learned a number of new terms. The first was mutation, which is a variation in the genetic code of an individual. Then we looked at the different kinds of mutations, starting with nucleotide mutations. We saw silent mutations that didn't affect the protein synthesis, missense mutations that introduced a new amino acid to the peptide sequence, nonsense mutations that stopped the synthesis of the protein midway through, and frame shift mutations that caused a complete change in the coding of amino acids and stop codons. Then we defined chromosomal mutations and looked at deletion, duplication, inversion, insertion, and translocation mutations that affect entire segments of the DNA sequence. We defined leakage as the flow of genes from one species to another, and saw that the gene pool consists of all of the alleles within a species. Finally, we looked at genetic drift and how it randomly causes changes to the gene pool and the founder effect, which is an extreme example of genetic drift. Thank you for watching this video, and good luck studying for your MCAT.